In this second video, again, we're going to be digging into this idea of transcription being regulated, dependent this time on how tightly packed the DNA is around the histone protein. So remember that in eukaryotic DNA, we have this idea of supercoiling. We have these histone proteins which allow the DNA to coil around itself, and we've got between individual nucleosomes, but this is a nucleosome here, this is one nucleosome. Between nucleosomes, we've also got the DNA being pulled together by various histone proteins. This allows the DNA to be very tightly packaged. When it's tightly packaged or supercoiled, we also need to appreciate that there's a certain amount of protection, because it means that environment can't impact the base sequence. And obviously, if you change the base sequence, then what you do is you mutate that particular gene. So in eukaryotic DNA, we have this idea of DNA being supercoiled and organized into nucleosomes by association with histone protein. By contrast, in a prokaryotic organism, there's no histone protein. So we talk about prokaryotes as having naked DNA. It's referred to as being naked, not because it doesn't have a nuclear membrane, but actually because of the absence of histone proteins. So if we look at the structure of the nucleosome, just to remind ourselves from what we should know from 7.1, we've got an octamer of different histone proteins. Then we've got the DNA being coiled around the outside. This histone H1 allows then the different individual nucleosomes to be pulled more together to allow this idea of supercoiling to take place. What we also need to appreciate is that DNA carries the phosphate group, which is a negatively charged ion. And what that means is that it wants to associate with positively charged histone. And here, this comes from BioNinja. These tail regions of the histone are positively charged ordinarily. And positively charged means that what we have is the ability of a positive charge and a negative charge on the DNA being associated with one another, and that tightly packages the DNA here. Now, the positively charged tail is a consequence of the tail being methylated. So methylation of the histone here allows supercoiling to take place. However, if this undergoes what's referred to as acetylation, the addition of an acetyl group, what happens is that the tail regions now become much less positively charged, which means that you have a loosening of the DNA around here. This loosening of the DNA means that it's much more accessible for RNA polymerase to get in there. So this is going to be transcribed. So in the acetylated version of histone, we get easy access for transcription, whereas in the methylated version, in this one here, we've got tightly packed DNA. We've already talked about in previous units about this idea of heterochromatin versus euchromatin. If we look underneath a light microscope, you can see that there are certain dense regions. The dense regions are referred to as the DNA being in this condition here. So heterochromatin refers to the idea that the histone is methylated, which means that you get very, very tightly packed DNA and little or no transcription activity within the genes within this particular part of the chromatin. By contrast, we've got acetylated histone. If we've got acetylated histone, it means that the DNA is much more loose. It means it's much more accessible for transcription. And this DNA, or this particular type of chromatin, is called euchromatin. So euchromatin has got easy access to being transcribed, whereas heterochromatin is less transcribed. Now, by contrast, we can also have, as well as methylation of the histone, we can have methylation of DNA itself. Now, methylation of DNA produces markers called epigenetic tags, and the methylated DNA, a little bit like methylated histone, means that it's much, much more tightly packed, and therefore less transcription takes place if there are more methylated tags associated with the DNA. Now, what's interesting here, but just to underscore, first of all, before we move on, that methylated histone in your mind must be very clearly distinguished from methylation of DNA. So methylation of histone means that we get lots and lots of supercoiling. Methylation of DNA changes over the course of an individual's life. Methylation of DNA and this methylated tags associated with DNA also can differ from one cell to another cell within the same individual. So that's a difference here. But in both cases, it means that there's less transcription. This diagram shows you much the same thing, but we're talking here about methylating the DNA. So we're methylating the actual base of the DNA very often. This comes from BioNinja, and BioNinja I would encourage you to have a look at for 7.2, and it underscores this idea that as you go through life, the number of methylated tags on your DNA can increase. Now, the number of methylated tags, as I've already said, can change within one individual depending on the tissue that we're looking at, but it can also change in terms of one individual compared to another individual. So as you grow older, there tends to be an increase in the number of methylated tags within your DNA, and that leads us into a new field of biology, which is referred to as epigenetics. So epigenetics is looking in particular at how transcription differs as a consequence of these epigenetic tags, which are methylated DNA patterns. So it's not looking at differences in the genes per se. The genes don't change, but the number of methylated tags on the DNA does change. This changes over time. It will be different between identical twins, and it can be used in analysis to pick up certain clinical conditions. In Cognity, there's a very, very good section which talks about the analysis of one specific gene and the number of methylated aspects of that gene and its correlation with breast cancer. So go back and have a look at Cognity very carefully. But here what we're seeing is that hypermethylation of this particular gene seems to be an indication of a diagnosis of a certain type of breast cancer. So that's interesting for us to look at. What's also interesting in terms of epigenetics is this idea that it's impacted by the environment and the conditions in which we live. So the environment directly affects our transcription by virtue of these methylation patterns. And this can change because of diet, because of exposure to certain kinds of pathogens. It can also be exposure to stress levels. It can be exposure to certain particulate matter within the air. But what we do appreciate is that these methylation patterns can be, but not always, associated with certain kinds of disease. Interesting is that, as I've already said, the methylation patterns within the same individual changes between different types of tissue. But it used to be thought that when you produced eggs and sperm, you wiped off those epigenetic markers. So you basically reprogrammed the DNA so that it was a blank canvas. What's now known is that most of these tags are erased. So this reprogramming takes place to a large extent, but not entirely. Now that is very profound because that means that the experiences and the exposure to certain environmental factors that your parents had could indeed impact the epigenetic markers that you inherited. So you inherit the genes, but there are certain epigenetic markers that might be inherited as well. This is a very, very new field of biology. It's really interesting. And I've posted some links in the Google Classroom, which are very good for you to have a look at. This also, Learn Genetics, this link here, also very, very worthwhile to look at. But what you need for your course is not a real deep understanding of what epigenetics is, but rather what you need to be able to do is analyze patterns in these analyze patterns of methylation within the DNA and to come up with certain sorts of diagnoses or look for evaluative aspects of that. So in the next couple of slides here, you can see that there are different epigenetic markers. The green here is showing that we've got hypermethylation. The red is showing that we've got hypomethylation, very low levels of hypo. And we're looking here at chromosome number 23, I think. But as we go through these links, we're looking at different chromosomes, and we're also comparing different identical twins. From here, if you go through this at your leisure, the PowerPoint is posted in the Google Classroom as well. Okay, what you eventually end up with a certain set of deductions. So we're looking at different chromosomes, and it's comparing chromosome 17, chromosome 20, and it's comparing differences in these methylation patterns of these two different chromosomes with identical twins. And this is the kind of thing that you can expect for your examination. So again, this is an idea that transcription is regulated by acetylation or methylation of histone, and in addition, methylation of the DNA directly. So this adds to what we did in the first video, which looked at the idea that certain proteins can impact non-coding DNA to cause an increase or decrease in transcription.